Okay, hi everyone. Welcome to another virtual seminar, one of the GPC seminars on climate variability. Today, we are delighted to have Mara Freilich, who is an oceanographer and is currently an assistant professor of applied math and earth and environmental sciences at Brown University. Mara received her undergraduate degree from Brown University in applied math and then did her PhD in the MIT Hui Joint Program and then joined UC San Diego as a postdoc. Mara has received a lot of awards for her work, most recently the Scripps Institutional Postdoc Program Award and the Phi Award for Excellence in Oceanographic Research Graduate Student Best Paper Award. And we are very happy that she's here today to tell us about some really an interesting topic that is the fluid environment, life in the fluid environment, ocean turbulence, and the global carbon cycle. So before Mara starts, just a reminder, please, during the seminar, please feel free to enter your questions into the Q&A feature. And at the end of the seminar, there will be time to answer those questions. So with that, Mara, please go ahead.
Great, thanks so much. Yeah, and Pedram, you can also feel free to interrupt if there are any clarification questions that would be helpful along the way. Yeah, so thank you all for being here. I'm looking forward to a discussion in the Q&A afterwards and the opportunity to share this with you. So as Pedram said, I'm actually currently a postdoc at Scripps and will be starting my position at Brown this fall. Today, I'll be talking about ocean biology and biophysical interactions. And so the title I've given it, Life in a Fluid Environment, I'll be talking about the interactions between ocean turbulence, ocean ecology, and carbon. And so this image that I have in the background is actually a satellite image of North Atlantic plankton bloom. And it really shows a few of the key elements that I find really inspiring for my research. So you can see they're different colors, right? This white color is probably coccolithophores. This greener color is probably a different species of plankton. But you can also see that there's swirls in these patterns of plankton. And that is really indicating that there's a tight coupling between the biology and physics in the ocean, with the physics shaping the distribution of biological organisms. And so one of the things that I find really captivating and fascinating is trying to understand how then is ecology different? How is that affecting climate and global biogeochemical cycles? The fact that there's such a tight coupling between biology and physics here. And this is significant because about 50% of the primary production that happens globally happens in the ocean, with the other 50% taking place on land. And what's different between primary production in the ocean versus on land is while we have forests on land, for the most part, the primary production in the ocean is actually carried out by microbial organisms. So single-celled organisms that cannot swim faster than the ocean currents. And so they really are transported and shaped by the currents. And we call them plankton, with planktonic organisms, meaning that they can't swim faster than the currents. And so really are drifting with currents. And so Pedram gave a really nice introduction. I also asked that I talk a little bit about my career path and my path into physics of climate. And so I won't repeat my whole CV, but just to say that so after an undergrad in applied math, I was really interested in applying mathematical concepts towards this mission of addressing societal problems using mathematical tools. And with a broad mathematical training, I was interested then in problems in ecology, problems in ocean physics and climate, and looked for opportunities to get truly interdisciplinary training and to do interdisciplinary work. And so I took advantage of an opportunity to do a Fulbright fellowship in theoretical ecology at La Universidad Católica in Chile, where I had the opportunity to work with Pablo Marquette and Rolando Rebiero there. Then I did my PhD in physical oceanography at MIT Hui Joint Program, advised by Amla Mahadevan. I'm currently studying oceanography as a postdoc at Scripps, working with Sarah Gilly, Matt Maslow, and Peter Franks here. And then I'll be a professor at Brown University. And I want to emphasize, although you see these institutions on the CV, what really matters, I think, throughout this is the people who I had the chance to work with at each of these institutions. And so that would be my advice for everybody, I guess. First, pursue interdisciplinary opportunities, think broadly across disciplines, but also really try to work with people who you enjoy working with. And so today, what I'll be talking about is some background information on climate as a complex system, focusing on carbon cycle feedbacks. Then I'll talk about how do we put this into a global framework in terms of Earth system models. Then I'll talk about some of my recent work on emergence of nonlinearities in biophysical interactions, and using data to understand how to better model ecosystems. And so I think it's well established that the climate is a complex system. The Nobel Prize in Physics in 2021 was for complex systems, with half of it going to climate science. But I want to get specific about what that means. And so one definition of a complex system is that phenomena arise from emergent interactions of component parts. And so interactions here is really key. And take this quote from theoretical ecologist Richard Levin, saying the truth is the whole. And so taking these two together, what we're really saying is that while we usually, for scientific understanding, we need to reduce a system to its component parts or make some simplifications and reductions. When we're working with a complex system, we have to understand that there may be emergent interactions, and that the phenomena of a reduced system may not actually resemble the phenomena of the full system. And one of the key things that can happen in a complex system is that there can be feedbacks. And I'll focus on feedbacks, and in particular in the carbon cycle here in this talk. And so what are some of those component parts that interact in a climate system? You have atmosphere, both dynamics and chemistry, ocean and marine ecosystems, cryosphere, meaning the icy parts of the climate, human society, which is currently a large driver of climate forcing, and then land, so the biosphere and other processes on land. And so that's at the global level, where we tend to think of global modeling, where I'm situated now, sitting in Southern California. If we bring this down to a regional level, we can see the way that these ecosystem processes and climate processes really have profound implications for human societies. With a large variety of processes. On the left-hand side, we have some of the more scientific processes, and at the bottom, this small component that I'll be focusing on of climate and ocean drivers of ecosystems, with profound implications for human societies, and there's many interactions then between the scientific understanding and the ways that we shape governance of these systems. Going back all the way to the traditional and ongoing stewards of this land, where I'm situated, the Kumeyaay, Cahuilla, and Chumash peoples. Okay, so I've been alluding to the ocean carbon cycle, and I want to give a definition of what that is. And so when we talk about the ocean carbon cycle, what we're talking about is all of the processes that shape the distribution and fate of carbon in the ocean. And so atmospheric carbon dioxide is exchanged through the sea surface into the ocean. And it's then taken up by the ocean through two main processes. One is by dissolving carbon into the ocean, and we call that the solubility pump. And so when carbon dissolves into the surface ocean, it can then be transported by ocean currents or by this overturning circulation. The other major process is what we call the biological carbon pump. And I'll talk a little bit more about what the biological carbon pump is, but the biological carbon pump is here biological organisms can then fix carbon into organic carbon, and there's a small leak in the biological carbon pump in this biological process that we call the biological carbon pump, where this fixed organic carbon can then sink down into the deep ocean, where it could be potentially sequestered. And so there's a robust and large natural carbon cycle in the ocean, with a large reservoir of carbon in the ocean. But the ocean is also taking up anthropogenic carbon. With that anthropogenic carbon, and so there are some feedbacks in this system. One example of a carbon cycle feedback relates to the solubility pump. So as carbon increases in the atmosphere warms through the greenhouse effect. When the atmosphere warms, then the ocean warms. When the ocean warms, carbon dioxide becomes less soluble in seawater. And so that means that as the climate warms, the ocean capacity to take up that increased carbon is reduced, and that's going to force more of the carbon to stay in the atmosphere, and this is what we call a positive feedback loop. So this, because then that will contribute to more warming. And so there are a large range of carbon cycle feedbacks. This is from the most recent, a graphic from the most recent IPCC report. And so here we have anthropogenic carbon emissions, putting carbon into the atmosphere, resulting in a wide range of climate effects, warming, but also changes in ocean and atmospheric circulation, and changes in the hydrological cycle. And there are important carbon cycle feedbacks both in the ocean and the land. And so in terms of positive feedbacks on the land side, we have soil respiration, permafrost thawing, erosion. You could go into more of the details of any of those if you want to ask in the questions, or look them up later. On the ocean side, I mentioned this decrease in solubility. There's also a few other changes in the ocean that may result in positive carbon cycle feedbacks that have to do with changes in the physics of the ocean for the most part. There are also some negative feedbacks, both in land and the ocean. Strengthening of the air-sea carbon gradient increases the partial pressure of carbon and can force more carbon into the ocean. On land, CO2 fertilization promoting plant growth. But then there's some where the sign is unclear, with a lot of uncertainty in the land carbon cycle of a number of processes where they could be quite important for carbon cycle feedbacks, but it's unclear. I'm going to focus on the ocean side because that's my area of expertise, and talk about the biological carbon pump again, which is one where the sign of changes in the biological carbon pump as the climate changes are not quite clear. And so let's dive into what the biological carbon pump is again in a little bit more detail. So here we have air-sea gas exchange across the sea surface. In the surface ocean, so a very narrow band at the surface of the ocean, about the upper 100 meters, with the ocean being about 4,000 meters deep, there's enough light for photosynthesis. And so in that layer where there's enough light for photosynthesis, we have phytoplankton photosynthesis occurring, but then there's a whole ecosystem where the fixed carbon is then respired and decomposed. And all of this happens because of the plankton at the surface, the bacteria, and these zooplankton, which eat phytoplankton and contribute a lot to the respiration. But as I mentioned earlier, there's a small leak in this cycle. So it's not a completely closed loop at the sea surface. About 10% of the carbon from globally from the surface layer sinks down into the deep ocean through what we call the biological carbon pump, rather than being utilized in the surface. And we call this carbon export or export production. This whole community at the sea surface relies on nutrient supply. So this ocean system, ultimately, it has to be mass conserving in terms of the elements, even though its energy source is light from above. And so as the similar to the way that carbon and nutrients sink downwards, they have to be carried upwards again. And how they're carried upwards is challenging. But for the most part, it would be by ocean physics. So physical circulation needs to be what brings nutrients back up again into the sunlit layer where they can be utilized by photosynthesis. And so the reason that there are nutrients then to bring back upwards is that there's this whole mesopelagic ecosystem that uses that carbon that sinks down from the sunlit surface layer. And so even though about 10% of the production from the surface layer leaks into the ocean interior, only about 1% of that will ultimately reach the seafloor where it has the potential to be sequestered in this geological carbon storage. And so the sequestration that takes place because of this biological carbon pump is a very small fraction of the total primary production in the ocean surface. Okay. So in this introduction, what I've talked about is climate as a complex system. And in particular, the carbon cycle feedbacks and the way that ecosystems and biogeochemistry contribute to this complexity. And so now let's make this a little bit more quantitative and talk about how we're going to model this complex system. And I'm going to introduce Earth system models here. And so Earth system models are climate models, general circulation models that also include these biogeochemical or biological components to them. And so fundamentally, what an Earth system model is, it has transport and forcing by ocean and atmosphere dynamics, so circulation, physics of the ocean and atmosphere system. We also have biological and chemical reactions, and then parameterizations, which are representations of physics that are not explicitly resolved in the model. And when we are building a climate model and Earth system model, we have to divide the Earth into a grid, and these grids have some discrete size that are much larger than the scales of individual molecules. And so the simplest model, or a simple model of a marine ecosystem, is what we call an NPZ model, with P being phytoplankton, Z being zooplankton, and N being nutrients. And we can then look at the exchange of material between these three components. And so here we have growth of phytoplankton depends on consumption of nutrients and light, and we can model light using physical principles of light traveling through water. Then we have grazing, where phytoplankton are eaten by zooplankton. And then phytoplankton and zooplankton, this is a mass conserving system here. Phytoplankton and zooplankton mass, or here we're using a nutrient currency, feeds back into nutrients through mortality and also sloppy feeding, where zooplankton don't eat all of the phytoplankton that they capture. And so this is a very basic model that represents the fundamental components of the system. Even in a model like this, if you add multiple nutrients and have nonlinear growth and grazing functions, you can begin to develop chaos using your periodicity, aperiodic dynamics, and even chaotic dynamics with a simple model like this that arises just because of ecosystem components alone, rather than any interaction between the biological system and an external environmental forcing. And so many Earth system models today use much more elaborate biological functions with many compartments. Here's an example of one that's used in a state-of-the-art Earth system model. The challenge as you add these levels of realism is that the models become increasingly computationally expensive because they have more and more tracers in them. And so with the limited computational ability, then there will be some choices that need to be made in terms of trade-offs. And so the power of these models is really how do we couple the biology and the physics together? And so here I'm going to look talk just about nutrients and transport of nutrients, right? And so here we have rate of change of nutrients is transport by an explicitly resolved velocity field. And then all of those functions I was just talking about before, these nonlinear biological functions, will just encompass on the right-hand side as changes in this tracer. And so if we then are representing this in a grid, we are not going to have every individual nutrient molecule or even a field of nutrients at high resolution. Instead, we are going to, in each grid, we'll have the average concentration of nutrient in that grid cell. The problem here being that these are nonlinear functions, and so the average of those functions will be different than the functions of the averages. And let's just look at this transport function. And so if we look at this transport function and expand that out, so this average transport, now we have three terms. We can assume that the velocity is incompressible, and then reduce this to just two terms. And so what we find is that the average nutrient concentration is the transport by the mean velocity. But then, oh, and I should have said, so what I've done here is I take nutrient and divide that into the sum of the mean nutrient, which is this overbar, and the nutrient anomaly, which is this nutrient prime. And same for the velocity. Velocity is equal to velocity, the mean velocity, and the velocity anomaly. Right, and so what we have is transport of mean nutrient by mean velocity, and then this term that's divergence of a nutrient flux that has to do with the covariance of these anomalies. And that term, this is what we call a turbulence closure, and representation of that term then is the topic of parameterizations in models. Because it's this irreducible nonlinearity of this system. We also have nonlinearities in this biological function that I haven't gotten to here, but that's a topic of research as well. Right, and so then the question is, what scale do we choose as our mean velocity and what scale do we choose as our prime? And these velocities are coming out of the Navier-Stokes equation, these equations for fluid motion. But those are multi-scale physics. As Joel Charney put it, the extreme generality whereby the equations of motion apply to the entire spectrum of possible motion, to sound waves as well as cyclone waves, constitutes a serious defect of the equations from the meteorological point of view. And so what he's really saying here is that there's no scale separation in physics. And that poses a challenge when we want to make this separation between a mean and a fluctuating component and come up with a turbulent closure scheme. And so ultimately, we usually end up for choosing what's the mean, what's the scale of the mean, and what's the scale of the fluctuations, really based on operational considerations. How much computing power do we have available to us to represent the processes that we're trying to represent? So this is showing climate models. This includes both general circulation models and our system models. And we're showing the on the x-axis, we have a year, so time going forward, and the horizontal grid spacing that those models had. And Earth system models are not going to be the highest resolution models here because of the additional tracer burden that they have from biogeochemistry. And so the finest resolution is getting smaller with time, but there's still quite a lot of physics that are not explicitly represented in our system models. And so from a biological standpoint, the processes that really matter the most or that have a large contribution are vertical processes, the processes that can bring nutrients upwards and that can bring carbon downwards. And although the equations really are the same at all scales, the dominant balances change. And so at the gyre scale, we have dominant balances between wind and gradients of Earth's rotation. Those are at the, so this is the rotating version, what we call the primitive equation. And And so those are typically represented explicitly in models. At smaller scales, the gradient in Earth's rotation becomes less important because that's a large scale phenomena. And instead, mesoscales is generally a balance between pressure gradient forces and Coriolis forces. Submesoscale is particularly interesting and exciting and is the topic of a lot of my research because vertical velocity becomes increasingly large at smaller spatial scales. But these balances then, and part of what makes it interesting and exciting is that these balances become much more complex. And we have to consider, like at the mesoscale, pressure gradient and Coriolis forces, but nonlinear advection becomes important, stratification, turbulent mixing processes can all, in various scenarios, play a dominant role in the balances. And so how do we parameterize this? I'm going to take a step back and just go with the simplest version, where we can parameterize these turbulent fluctuations, covariance between anomalies. Here I'm looking at covariance between velocity anomalies. And we can do a diffusive parameterization, or say that we have some diffusivity and we multiply that by the gradient of the velocity. And this goes back more than a century, where Taylor, in conversation with Richardson, who had made very careful observations of smoke coming out of a chimney with a mean wind, showed that with this, so you could look at the spreading of the smoke out of this chimney and choose a value for this diffusivity kappa that depends on the autocorrelation between the velocity anomalies here. Great. And what's interesting about this is that now we've moved from a closed system where we had a deterministic set of equations for an ecosystem, if we were to add this diffusive parameterization to an ecosystem, to now an open system where we're really talking about unresolved environmental processes and the way that they affect a focal ecosystem. And so it's a different way of thinking about how to derive some of the fundamental principles. Great. So to summarize, right, we've built an Earth system model that depends on transport and forcing by ocean and atmospheric dynamics, and the way that that couples to the biological chemical reactions and to parameterizations. And we can ask which of these matters the most. And so this is some work, if we look at global mean temperature over time, and we're trying to use some of these models to ask what will the global mean temperature be, say in 2100, right? One of the central things that you hear coming out of climate science. And we can ask which of these different processes matters the most, right? So climate is a chaotic system and that contributes to internal variability. We can see that that internal variability is not contributing most of the uncertainty at long time, or even really at short time when we're talking about global mean temperature. Then these model components, as I've been saying, there's some questions we have about the precise way to represent some of these physical processes that are below the grid scale of the model because we don't have the computational capacity to explicitly simulate the system. And that's this model uncertainty here. And so model uncertainty is part of the spread, but it's not the largest part of the spread again, right? The largest part of the spread in global mean temperature at 2100 is what we call scenario uncertainty. So that's how much carbon dioxide are humans going to put into the atmosphere, right? So that's something under our control and does not have to do with improving climate models. But the scenario is actually somewhat different if we look at biogeochemical processes, and in particular, if we begin to look at regional scales. And so at the global scale, here I'm looking at the air-sea carbon flux. So how much carbon is going into the ocean? Here's a map of what air-sea carbon flux looks like in a model. And here over time, this is results from a paper led by Nikki Lewandowski. What is the fraction of the total variance among Earth system models that has to do with internal variability? So at a global scale, that's fairly small. That has to do with the model structure. So at short times, that's actually quite large for Eric C. Carbon Flux, but at longer time scales for Eric C. Carbon Flux globally, again, it's scenario uncertainty. How much carbon are we going to be putting into the atmosphere? In the California current, however, model uncertainty really dominates all the way out to long time scales. And so if we want to have predictability at the regional scale, we need to gain more confidence and more certainty in the models that we're using. So while we're doing a really good job for these global predictions, I think one of the things that climate science is going to be increasingly called to do in the next couple of decades is to provide more regional predictions as societies begin to adapt to and think about how to mitigate climate change at much smaller spatial scales. And so here, I talked about the ways that biogeochemical fluxes are parameterized, focusing on this diffusive flux parameterization, but I should say they're much more complicated parameterizations that are used particularly for physics. And touch briefly on the ways that climate outcomes are sensitive to parameterizations, particularly at regional scales. And so now I'm going to talk about some of my recent work on emergence of nonlinearities in biophysical systems. And so mathematically, the types of equations that we focus on are reaction, advection, diffusion systems. And these can be used for ocean biogeochemistry. There's other climate processes like cloud microphysics, where reaction, advection, diffusion systems are a really relevant modeling framework. Here, I'm going to focus on just an advection reaction problem and bring diffusion back in terms of parameterization later on. So we're going to look at this very idealized scenario to help us understand some of the fundamentals of the process here. And so here I'm looking at evolution of nutrient concentration on a water parcel that's oscillating vertically in a water column. And so in the background, there's some equilibrium concentration of nutrient. And as these water parcels move up and down in the water column, we have the linear biological modification that restores the nutrients either quickly to the equilibrium concentration or slowly. And if the restoring is fast, then the nutrient on the water parcel is very similar to the equilibrium concentration at any point on its trajectory. And as a result of that, the nutrient anomaly is very low. By contrast, if the restoring is very slow, then the water parcel maintains a kind of average concentration on its whole trajectory. And the nutrient anomaly is then large at either the top or the bottom of the trajectory. And so we see here that the time scale could have an important effect. These biological reactions have an important effect on the nutrient anomaly. And if we frame that in terms of a nutrient flux, so here we can come up with an analytic solution if we specify a vertical velocity. So we specify a vertical velocity, just a sine wave, right, that oscillating with some frequency, and that frequency here is omega. And using that linear biological reaction, we can actually explicitly solve for the vertical nutrient flux, W prime, N prime. And the equation we get is a nonlinear function of the biological rate. And so this is really interesting because we had a linear biological function, right? We linearized biology, really simplified it as much as we could. But flux is an inherently nonlinear process, as I said earlier. And so that gives us this nonlinear function when we average over time on this trajectory. And it's even more interesting because for a given vertical velocity frequency omega, the flux is actually maximized when the biological uptake rate lambda matches that flux. And we can understand this by thinking about the phase relationship between the nutrient anomaly and the vertical velocity. So for a given vertical velocity, if we have fast uptake of a nutrient, we have a positive flux over the whole trajectory, but we have a very low nutrient anomaly. If we have slow biological uptake, we have some region, we have some times where we have positive flux and sometimes where we have negative flux, but the average of this will be very small because the positive and negative flux cancels out. And then the optimum is somewhere in between, where we have a large enough nutrient anomaly that we have large flux, but it's in phase enough that the integral can still be positive. I saw somebody raise their hand. I don't know if there's a question. No, okay. And so one of the tools I use, and so that's a kind of very simple theoretical justification. Vertical velocity in the ocean is a lot more complicated, has all of these multi-scale processes. And so one of the tools that I use is primitive equation models here, solved using a finite volume solver. And so this is, we take a patch of the ocean and make an idealized model of just a patch of the ocean with a realistic vertical velocity. And this is a snapshot of that model run. And I couple to this a restoring biological model. So the same restoring model we have. So we're still going to be using this very idealized biology. And I use a range of biological rates, lambda, on trajectories from this model. And so if we look across the range of just the trajectories alone, we could actually model a nutrient flux in terms of this diffusive flux that we got from Taylor. And so we can say we have some diffusivity that's the autocovariance of the vertical velocity. So now we're looking just vertically instead of horizontally, the way with the smoke plume, but it's a similar model. And that gives us a diffusivity in this range here, depending on where we are in these complex physics. Diffusivity could be slightly different, but it doesn't depend on the biological rate at all. But when we do simulations with a wide range of biological rates, similar to the theoretical case, what we have is a nonlinear, vertical nutrient flux is now a nonlinear function of the biological rate. And the only modification that we need to make to this diffusivity is that it needs to account for the biological timescale lambda. And so there's a weight, so now we have a weighted vertical velocity autocorrelation is the solution for this vertical nutrient flux. And so this memory effect is actually one of the hallmarks of a complex system, is that because of these emergent interactions, a sort of memory arises. But then the question was, so this is all in a model, is this relationship observable? And I think this is one of the challenges we have, is bridging between models and theory and observations. And so one of the ways I address that is actually by going to sea and trying to test models in the theoretical framework. So here's a picture. I was working with collaborators in Spain. And we test these theories by taking water samples. That's what I'm doing from these gray bottles. They're called Niskin bottles. And then we also use a wide range of particularly optical sensors. Then we would lower this whole instrument into the ocean to take some key measurements. The observations that I'm going to be using to test this theory are from the Hawaii Ocean Time series, where 30 years of incredible effort by a large community of people have contributed to collecting these observations. And so from that, we can synthesize across these 30 years of observations and look at the carbon-specific growth rate in the surface layer. So that gives us a range of growth rates. One thing that's really nice about this, and I didn't point out the numbers on the previous slide, but these growth rate ranges are really close to the optimum value. The optimum value that we had from our idealized model was about 0.3 per day. So using realistic physics, and those are really realistic values for growth rates in the ocean. So that's nice. But getting observations of anomalies, getting observations of vertical velocity is challenging. Getting observations of nutrient anomaly is less challenging. And so what I'm going to do here is to say that this W prime N prime, which here is the same as W prime C prime, is euphotic zone integrated productivity, relying on actually our linearized theoretical model. And if we do that, we can then see that we get an emergent nonlinear relationship again, where our estimated nutrient flux is a nonlinear function of the carbon specific growth rate. And this is actually really nice because even though, as I said earlier, we think that physical processes have a strong role in controlling biological productivity, some recent work had found that kind of counterintuitively, primary production in this region near Hawaii actually did not seem to be have a linear correlation with the nutrient anomaly available from physical forcing. And that was counterintuitive, but the reason, as this theory shows, could be that there's a nonlinear relationship. And this theory provides us a really nice framework to analyze specifically why and when that nonlinear relationship should arise and the implications of that. And so I pointed out that now we'll go back to the model again. And one of the things I pointed out at the beginning was that we have this range of trajectories that have different vertical velocity characteristics associated with them. And so if we map those in space, what we find is that rather than having an optimum biological rate that's around 0.3 per day, instead we have a range of biological rates that range over a couple of orders of magnitude. And so these are all actually fairly realistic in the ocean, with some slower growing organisms like cyanobacteria would be on the smaller end and faster growing organisms would be on this larger end. And this mechanism of interactions between biology and physics could provide a coexistence mechanism where on these very short scales, we have coexistence of communities with very different emergent growth rates. And so that's another thing that more recently, I tried to go out into the field and test these are results that just last October, we went out to see and did some surveys across small scale features. And we found that there was quite a lot of difference in at least photosynthetic activity on small scales. And we're still working on what the biological rates are from that and what biological communities are associated with these rates. And so what we found here is that there's a growth transport feedback where nutrient, carbon, and oxygen fluxes, here I gave the case of nutrient fluxes, but this framework could really be extended to other biogeochemical tracers, depends on coupling between biology and physics. I showed how nonlinear fluxes can emerge from linear models and talked about the ways that variability in vertical velocity characteristics on water mass trajectories could be a biodiversity generating mechanism in the ocean. Okay, and so now before wrapping up, I'm just going to touch briefly on using data to find model functional forms. How can we move beyond this sort of linearized framework while maintaining this close relationship between theory, models, and observations? And so here, returning to our reaction advection diffusion problem, I'm now going to account for some of the ways that organisms may interact with each other by using nonlinear biological functions. Here, looking at interaction between growth and grazing. And I'll do this in a more diffusion reaction system, which diffusion reaction systems are known for emergence of pattern formation processes and things like that. I will not be focusing on that here, and instead really just focus on the model functional form itself. And so this work was inspired by observations, autonomous observations, again, large oceanographic community efforts compiled by Alex Mignot. And what he observed, so here we're looking at time over the year. And so phytoplankton are known to bloom in the spring, right? Similarly to the flowering of plants in the spring. And so if we look at this blue line, we see that phytoplankton concentration peaks in the spring. But in the ocean, the surface layer, the surface mix layer deepens a lot in the winter. And so if we actually integrate all the phytoplankton biomass over the surface layer, we find that the biomass cycle has less variation, and there's actually increase in phytoplankton biomass over the winter time. And one mechanism that's been proposed for this is that when there's a shallow mixed layer, zooplankton are able to control phytoplankton populations very closely. But when the mixed layer deepens, phytoplankton are diluted, and the encounter rates between zooplankton and phytoplankton decreases. And so a less technical way to say that is phytoplankton or zooplankton, which eat phytoplankton, are just having a harder time finding them. And so if we take a very simple model of plankton dynamics, this is like the NPZ model that I introduced earlier, where we have a balance between growth and grazing. Now I've put nutrients just as an implicit term. Because this is a mass conserving system, we can say that nutrients are just the remainder here. But what I've added is a dilution term, where when the ocean surface layer deepens, we dilute both phytoplankton and zooplankton with water from below that didn't have any biological organisms or very low biological concentrations before. And so what function we use for this grazing function, GP, we thought would be really crucial. And the type of grazing functions that are typically used are linear, and then saturate. So say zooplankton eat phytoplankton, but then there's some, they can't keep eating at the same rate when there are enough phytoplankton around. But this emergent phenomena that had been observed in the winter time, where dilution results in an increase in phytoplankton, what we argue relies on an inflection, where at low phytoplankton concentration, phytoplankton find a refuge from grazing. And so this nonlinearity sorry, there's a back. Or this nonlinearity in the biological processes ends up being really important. And so there's some algebraic analysis that we go through, but essentially, if we take these three functional forms for grazing and look at dependence and we deepen a mixed layer, we find that with a linear function, so we have here mixed layer depth on the vertical axis and phytoplankton biomass on the horizontal axis. We find that phytoplankton are able to grow when the mixed layer is shallow, but when the mixed layer gets deeper, then they don't have enough light to grow. The average light goes down, and so then it's loss dominated. The phytoplankton are not going to keep accumulating. But if we have an inflection at low phytoplankton, what we find is that there is a regime where if the mixed layer deepens enough, the phytoplankton get this release from grazing and they're able to continue growing, even in low light conditions. So they'll be growing very slowly, but because this balance between loss and growth goes the other way. And so inspired by these observations, then we try to fit models to observations. And what we find is that we're able to fit, so these are idealized models, they don't fit the observations perfectly. But so this green line is with a type 3 functional response. We see we are able to get positive net population growth in the winter time. This orange line, on the other hand, is the type 2 functional response, so saturating functional response, and more linear. And this, even no matter what parameters we give it, it can't result in this wintertime accumulation of phytoplankton. And so I think there's a rich area here where you could do much more complicated equation fitting or equation discovery frameworks using this data that's available from biology, and really to help figure out what sorts of functional forms should we be using. with this grounding in theory of why those functional forms are giving us these emergent outcomes that we want and that relate to the observations. And so what we found here is that the observed annual cycle in phytoplankton can be used to infer grazing dynamics. And that's really significant because we have just less observations of grazing than we do of phytoplankton distributions and growth. And this wintertime growth could have implications for the spring bloom period as well, particularly in a changing climate. And so in conclusion, the message that I really want to take away is that these nonlinear dynamics mediate responsive biosphere to climate and environmental forcing. And that as we're parameterizing climate models, we need an understanding at the fundamental level of how biology couples to physical processes, whether it's advection or these turbulent boundary layer processes that I talked about in the second half, because these result in emergent dynamics. And the way forward here is to increase predictability, particularly at regional scales, by synthesizing theory, models, and observations. And so thank you everybody for coming. And I put my contact at the bottom.
Okay, great. Thank you very much for this fascinating talk. So I asked the audience to please type your questions in the Q&A box. But while you are doing that, I wanted to, actually, Albion reminded me to mention that Samara is a member at large of the executive committee of GPC. I put the website in the chat room. Please check it out and get involved if you're interested. Also, as you know, all of these seminars are recorded and we share the recording on our seminar website that I'm going to post the link in the chat. So we already have one question from Matthew. Mara, you can see the question, right?
I can.
Yeah. Okay, go ahead.
Yeah, so the question is, in the context of nutrient, carbon, ocean feedback, what is your idea on ocean fertilization as a climate change mitigation solution? My answer to that is I think it needs to be approached extremely carefully. So as I pointed out, ecosystems are incredibly complex. And if we fertilize the ocean, we are going to be, I mean, the idea with fertilizing the ocean is really to perturb ecosystem dynamics. And I think that could be, it could result in very unpredictable outcomes. Because as I showed, we have very limited predictability of ocean ecosystems.
I have a question, Mara. So I'm wondering if you can give us a sense of the timescales of these vertical transports of carbon versus basically the biology versus heat and momentum, and how much these would vary regionally in the ocean.
Sure, right. So one of the important takeaways from the results that I showed are that the coupling, that these biological reactions decouple transport of biological tracers from heat and momentum, right? And mean that their transport can be slightly different. And so if we have a parameterization that works perfectly for heat and momentum, it may not necessarily work for biology. Right. And so in terms of some of the timescales, there are a wide range of timescales, right? So the timescales of biology, as I said, are this kind of 0.01 per day to 1 per day. I think one of the important outcomes of this work, and this is more on the technical oceanographic side, is that while a lot of the focus on the importance of sub-mesoscale dynamics has been on the magnitude of velocity, so saying that the magnitude of vertical velocity increases to about 100 meters per day at sub-mesoscale, what I showed is that what really matters is actually the frequency. And so understanding the spectrum of frequency then becomes increasingly important. So that's something we have from models, and right, we don't have a scale separation, and that's why I'm kind of hedging on this, right? We don't have a scale separation, so I can't say like what is the one frequency of vertical velocity. But the frequency spectrum is a red spectrum in the ocean, but the details of that, I think, are a topic of current research, right? What actually is the frequency spectrum of ocean vertical velocity?
Hey, thanks. There are a couple of more questions in the Q&A.
Okay. Are you modeling vertical transport as uniform within each model cell? So yes, it has to be, right? So the model cell, we have a flux through the boundaries, through the top and bottom boundaries, where we need to choose one vertical flux to go in the top and bottom, but the role of the parameterizations is to use the information that we do have at the core scale, then to look at the outcome of the heterogeneity that we know exists within the model grid cell. And then the other is, does the presence/predominance of critters cause local upflow or downflow? Yeah, so I'm going to take that two ways. So one is zooplankton, oceanographers like to say, have perform the largest mass migration on Earth. So they'll migrate up and down to about 1,000 meters, or potentially to 1,000 meters, depending on the species, every day. And that actually can be a carbon transport mechanism itself, because they respire, right? So they've eaten at the surface, but then are respiring subsurface. Another, I think another thing that that this question may be getting at is, do these organisms affect the flow? And so ocean organisms certainly are affected by high Reynolds number dynamics locally around themselves, right? And so the way that they experience the world is much more viscous than our experience of being in the ocean because of their small size. And so the flow around these organisms can actually be significant for their ability to feed and interact with the ecosystem. Whether these organisms affect the flow itself, there's some suggestions that they might, particularly in like near coastal areas where there are very high concentrations. But on the whole, they're likely not a kind of leading order in terms of mixing the ocean. Or definitely not leading order in mixing the ocean, but in terms of flow dynamics. Could you maybe comment on how ocean acidification fits into this? For example, changing growth times, et cetera. Yeah, right. So one of the really important consequences of this air sea carbon flux is that as there's more carbon in the ocean becomes more acidic. That can have a lot of ecosystem consequences, right? So it could change which organisms can survive in the ocean. On my title slide, when I showed the kind of white swirls, those are coccolithophores. They have these calcium carbonate shells, and their ability to precipitate them could be dramatically reduced if the ocean, or as the ocean becomes more acidic. And so as you change the composition of ocean ecosystems, then you do change the net ecosystem growth rates, but also just all of the complex biogeochemistry that I glossed over, but alluded to as being important for Earth system models.
Okay, so can I ask one more question? So on one of your slides, you showed the importance of memory, right, in calculating the flux. But is that currently accounted for in parameterizations? Because parameterization for like momentum and heat are often like kind of assume like instantaneous equilibrium. But I guess these time scales that you are talking about can be long. So do the parameterizations account for that?
No, I don't think, right? So it really depends on the type of parameterization is going to be used, right? So the details differ, but on the whole, most parameterizations, which rightfully are focused on getting the energetics of the ocean correct, may not be appropriately representing flux of biogeochemical tracers in particular. And part of that then has to do with these memory effects on trajectories. Yeah, so I think it's pointing out that a biogeochemically focused parameterization may help with representing ocean processes, but it will at times, for multiple reasons, be different than what you need for heat and momentum.
Okay, great. So it seems that there is no other question. So again, thank you very much, Mara, for this really interesting talk. And so the recording will be posted on our website very soon. So if the audience want to watch it again, or if they have more questions, reach out to Mara. So thank you very much again, Mara.
Yeah, thank you. Thank you all.